first webinar of three. So this is the North Thames Genomic Medicine-led webinar for the genetic testing for patients with cancer with unknown primary. So this is specifically for clinicians from North Thames, East and South West, because you'll be routing your liquid biopsy samples into the Marsden. So the other two GMSs that also do liquid biopsy testing will be doing separate webinars for their respective GMSs. So I'm joined today by colleagues from Manchester and the Marsden who are going to run us through, do you want to just flick to the next slide, Fiona? She's going to run us through this agenda where we're just going to talk briefly about why genetic testing for CUP, and then the Marsden is just going to take us through in terms of the logistics of how to order biopsy testing, because it may be new for some of you on this call. And then there's a brief section at the end in terms of other educational resources. We are going, as I said, we are going to record this, and hopefully we'll send out the slides as well, and they'll also be located on the CUP NHS Futures pages, which Natalie Cook will go through at the end. And we're going to do this for about 25 minutes, and then we'll have some time at the end for Q&A. Please use the chat function for that, and then we'll moderate that at the end. So if you do find yourself not from one of those three GMSs, there are some QR codes at the bottom here in terms of signing up for the other regions webinars. So I'm going to pass on to Natalie, Dr. Natalie Cook, who's going to lead us through the first section of the presentation. Thanks, Natalie.
Great, thanks, Paul. I'm going to request control if that's okay, so I can go over my slides. Hopefully that will work. I'm going to talk to you. So my name is Natalie Cook. I'm a medical oncologist at the Christie Hospital, and I am the current chair of the UK CUP group. So we've been working for quite a long time now, trying to basically show that our patients diagnosed with cancer unknown primary should have genomics integrated into standard of care. I'm going to show you a little bit of evidence from some of the studies that we've run. There have been big global studies like Capisco, which was sponsored by Roche. We've got our investigator studies like CupComp. And actually, we know now that if we integrate this next generation sequencing in tissue or blood, it can improve survival. We've also looked at the costings of the CUP pathway, which we know is very expensive. We know we have these genomic assays readily available, and looking at a health economic analysis, we know it's cost effective compared to standard of care. So with the work that we've done and others, basically, we have had this case approved now after submitting to the test directory, and so we now have to try and implement this, and that's the whole point of today, looking at the logistics, how we can ensure everyone has the same access across the whole of England, and then obviously how we then interpret the results. So I don't have, so can we just go to the next slide? I think, is it me? Have I got some? We know that our patients need rapid referrals, they have rapid appropriate investigations, and really time is of the essence with these patients. But what we haven't had until recently is access to the more comprehensive molecular diagnostics. So whole genome sequencing has been made available on the test directory from around April 2022, but because of the need for a fresh tissue biopsy, there's been quite limited uptake in certain areas of the country. Not all, but we know that this has been a barrier for patients having access to the genomic sequencing that they need. And the issues with tissue is that obviously our patients have had delayed pathways before they get to see us. Most of the biopsy samples are put in fixed formalin, which makes it much more difficult to do whole genome sequencing, although we do have RNA later now as well. And often the tissue is exhausted because of all the rounds of immunohistochemistry you have to try and get to a diagnosis. Patients need urgent treatment, so there is reluctance there for having repeat biopsies and waiting for the results of these. And until recently, we've had lack of integration into standard of care. So now we're pleased to announce that from next week, the 13th of July, NHS England will add liquid biopsy and tissue testing onto the genomic test directory for CUP. So this is alongside whole genome sequencing. So we have different options for our patients. So this is great news because we know that not one size fits all with these patients. So we do need these options available. And these are the codes that will be used. And again, the test directory will be updated from next week. So just to go into a little bit more detail about the actual guidance and the eligibility criteria. So for liquid biopsy, this is going to be available for patients that meet the following criteria. So ECOG performance status 0 to 2, so those patients that you expect to potentially go onto a treatment option. They have to have a diagnosis of cancer or carcinoma of unknown primary origin as per our NICE and ESMO guidelines, which people that treat in CUP will be familiar with. And because the liquid biopsies can be affected by patients on treatment, they have to be off treatment to have the ctDNA. And they also have to have had a discussion at a local CUP MDT confirming the diagnosis. At the moment, we're not including patients with a malignancy of unknown origin or a non-epithelial, non-neuroendocrine malignancy, because these are not in the guidelines. And at the moment, for these patients, the malignancy of unknown origin, whether you haven't had a biopsy, we don't have the evidence to say that this is going to be an effective indication. We will obviously be looking to revisit this in the future. So for those of you that may not be familiar with liquid biopsy testing, so we know that cancerous tumours can release fragments of DNA into the bloodstream, known as circulating tumour DNA. And it's through these kind of liquid biopsy testing in the blood that we can identify key genetic drivers of cancer. So within the NHS, and some of you may already be familiar with this, we already have liquid biopsy testing available for patients with suspected stage 3 or 4 lung cancer, and also ESR1 testing for specific advanced breast cancer patients. Plus other genomic tests as well. So it's great we already have these indications, and we've been working really closely with the NHS circulating biomarker networks of excellence to try and look at CUP as the next indication. So a real thank you to those for leading through this and helping with some of the health economics analysis as well. And so just going on to my liquid biopsy testing. So essentially, the research that we've been doing has shown that ctDNA testing can improve the diagnostics and management stratification of patients with CUP. So we're just about to publish a study called CUP-COMP, which should come out in the next 2 weeks, which basically shows that out of all the patients we profiled, 2/3 had actionable alterations detected, and actually just genomics alone could help refine a primary tumour type in about 20 to 30% of cases. And Edge Health, working with the circulating biomarkers networks of excellence, did some health economics analysis with us about the use of ctDNA in the CUP pathway. And actually it shows that despite the fact that this is an added test, this could actually result in a net impact of cost savings rather than it costing the NHS significant amounts of money, which has led to then this approval going through for the test directory. And so we know that some patients may be on treatment or they may not be able to have a ctDNA test, a liquid biopsy test. So we still think it's important for these patients to have access to tissue testing if needed. So to have tissue testing, patients must meet the following criteria. So same with the ECOG performance status, these patients have to be ones that you're thinking about treatment for. They have to have a diagnosis of carcinoma unknown primary as per ESMO NICE guidelines, and discussion at a local CUP MDT. And importantly here, we have to have adequate biopsy material available for DNA extraction. And obviously this is something you'd have to liaise with the pathologists at your local hospitals about. But still, we're not able to obviously do any of the non-epithelial tumours as part of this, as they're not part of the guidelines. And whole genome sequencing will still be available. So obviously these are the current guidelines that we have in the test directory. There needs to be fresh tissue there available for DNA extraction. We perhaps would be looking at this if we don't have any informative results from ctDNA or tissue, and therefore whole genome sequencing may be the only additional way of getting actual information or potential suggested tissue of origin. And then these are the current genes that we'll be testing, and we are looking to add to this all the time because we know that the more genes we can look at, the better in CUP, because CUP could be any number of different cancer types, including hematological and sarcoma type malignancies. So the ctDNA panel is here, and the tissue panel is here, and obviously the red targets on here are also clinical trial targets where we can match to potential clinical trial options as well. I'm not going to go in any detail, but this will all be updated on the test directory from next week. So it's really exciting we have all these genes included, and again, we will be looking to even increase the number of genes we can test going forward. And then the final slide I think I have is just some kind of guidelines. I'll come back to John's question later. I just saw it pop up. So the guidelines we're looking to, now this is kind of a work in progress as well, because for some of you, this will be the first time you're looking at these types of tests in CUP, and it can be quite complicated without knowing the tissue of origin as well. So our plans will be to look at ctDNA or tissue, depending on what's been requested. We expect probably about 70 to 80% of patients will have ctDNA tested first, but around 20 to 30% maybe tissue. We would advise, certainly for patients, as you're starting getting going with this, that these but cases are discussed in your local genomic tumour advisory board, and we'll come back to that later. And there may be different actions of the result of that. It might be that actually, unfortunately, the ctDNA has not informed any potential actionable targets or even a tissue of origin, and therefore you'd continue to manage this patient as per CUP and as per your local guidelines. It may be that actually you have got some potential actionable targets that could mean the patient is eligible for clinical trial options, and these could be identified as part of the GTAB discussions. And it may be actually the pattern of genomics could be suggestive of a potential tissue of origin. And certainly we would ask then that their patient is rediscussed at the local MDTs, either the tissue of origin MDT or the CUP MDT, to redefine the tissue of origin. And this is something that we've been doing quite often in our clinical studies now. If the ctDNA or tissue results are inadequate or failed, then obviously you could look at the other options, so either doing ctDNA or tissue if they meet the eligibility criteria for the repeat testing. So this, again, I'm happy to take comments on this, but this is the current kind of guidelines for how we would roll this out in CUP. And again, this is kind of new to a lot of you, so please please feel free to reach out if there are any questions or comments off the back of this webinar. So I think I can hand over now, because I think we go over to the operational side. Thank you.
Thanks, Natalie. Fiona, do you want to flick the next slide, please? Sean, do you want to take over?
Yep, so I'm just going to go through the process for collecting a sample, sending a sample to us, and the laboratory workflow, and then interpreting some of our reports. Just have the next slide, please. So the process is already in place for lung and breast cancers, and CUP will follow the same pathway. So blood collection kits need to be purchased by individual trusts, and we recommend ordering these from Alpha Laboratories. And the kit that you'll receive will have 2 Streck cell-free DNA tubes, a Royal Mail tracking label to ensure the samples come back to us, a package to send the sample in safely, a biohazard bag, and an absorbent pouch. Next slide, please. So as well as ordering the kits, the Royal Marsden will send out some documents to support the ctDNA requests, and these documents will include an instructions for use that provides some simple instructions on how to collect the sample and send it back to us, a frequently asked questions document, and a test request form that is specific for CUP. If at any point you have any queries regarding these forms, please contact Marsden360 at rmh.nhs.uk, and somebody from the team will be happy to help. Next slide, please. So the next few slides, I'll go through some of these documents in more detail. Firstly, there's a specific form is being developed for CUP. It's in development at the moment, but it'll be very similar to this form. And what's important is that it's completed fully and returned with the sample, and that the correct form is used. So can you please ensure the date the sample was collected is written on the form? There'll be a Royal Mail tracking number provided with the kit. Can this please be added to the form? And we also advise that you make a note of this locally. So if there's any issue, then we can track where the sample is in the system. Please tick the patient eligibility box to confirm the patient meets the eligibility, and add any relevant clinical information such as tumour histology or suspected primary site, because this can help guide interpretation of the results and our report comments. Please also inform us if the patient's had a transplant or transfusion. And then on the report, you need the details of the ordering physician. We need the email address and the email addresses of any other people who are going to receive the reports. Need to be the secure email such as nhs.net or your hospital email. And finally, the form must be signed by the consultant or an authorised delegate such as a CNS. Next slide, please. So this shows how about collecting the blood sample. So the tubes provided in the kit are Streck tubes, and these are the only tubes that will be processed. So please don't use any other tubes. And these are glass tubes, so care must be taken when handling these. Before collecting the sample, ensure that there's no cracks in the tube and that they haven't expired. Then collect the sample by normal venipuncture, and 21 or 22 gauge needles are advised for this. And ideally, fill each tube with 10 ml of blood. Once the tubes are filled, mix gently by inverting them 8 to 10 times. Please don't shake them because this can compromise the sample. And it's important that the tubes are kept at ambient temperatures. They're not refrigerated or frozen at any point. And then it's important that the tubes are labelled with the patient name, date of birth, hospital number, and collection date. And because any unlabelled tubes cannot be processed. Next slide, please. This is the instruction for use document that will be emailed out to you. I've already gone through the blood draw instructions, and I'll just briefly go through how to package a sample to send to us. So once you have your labelled blood tube and your request form ready, package the blood tube in the absorbent pouch and place in the biohazard bag, and then you can go into the box. And the request form also goes into this box. Please ensure there's only one collection kit per patient, so please don't put any other patient samples into the same box. Once everything's in, it can be sealed and then taken to the post room to return to the Royal Marsden. Ideally, samples should be sent as soon as possible after they've been collected, and then we would be receiving them within 48 hours. It's recommended not to avoid taking on a Friday if possible, but if it is unavoidable, then please post on the Friday rather than delaying posting. Next slide, please. This slide is just to summarise the process that happens once we've received the samples. The samples are booked in at the sample reception, and the details on the request form and the blood tubes are cross-checked. Any unlabelled samples will be discarded, and you'll be notified the sample has been rejected, and a repeat will be requested. Also, if there's any information missing on the request form, this may need following up, in which case you'll be contacted, but bear in mind this can delay starting processing, so please complete the forms fully. Once the sample is booked into the EPIC system, it will proceed to plasma isolation. At this step, it's done as soon as possible, really, after we've accepted the sample. The plasma is separated from the whole blood by a double centrifugation step, and then the plasma layer is removed, and this contains the cell-free DNA. The cell-free DNA is extracted and quantified, and this process takes about 2 to 3 days. Following this is the next generation sequencing library prep. This is a very automated 3-day protocol, and at the end of it, enriched libraries are sequenced on the NextSeq, and sequencing takes about 24 hours. Following this, the data goes through the Guardant bioinformatic pipeline. This is a custom developed pipeline that filters the data and does several QC checks, and this runs for about 12 to 14 hours. And after this, the data is available to us for analysis and reporting. So 2 scientists analyze the data and interpret the variants. Decision support tool is used in the workflow to add to the scientist's expertise and to try and streamline the reporting stage so we can report as quickly as possible. And this software is also used then to generate a custom design report. And this analysis and reporting is done in around 24 hours. And this patient report is emailed out, and results are normally reported in around 10 days. Next slide, please. So for the next few slides, I'll show some examples of some reports. So the reports generated by the software can be quite long and complex, but we have summarized the main findings on the first page of the report. So at the top of the report, you'll see the overall comment section. This is a summary of the results and the clinical scientist's interpretation of the relevant variants detected are all described in this comment. Underneath this, clinically relevant variants are listed with a nomenclature, the variant allele frequency, and the pathogenicity and actionability assigned by the scientist based on the current guidelines. The assay can also detect high microsatellite instability, which may guide treatment decisions. And this is, in this example, MS high was not detected, which is that comment at the bottom. The initial page then often has all the key information is needed to contribute to patient management, but there's a lot more information in the report further down in the main body of the report. This is an example of a CAP report with potential actionable variants detected. You see a BRAF V600E variant being detected, which is a driver variant occurring in multiple tumour types such as colorectal, lung and thyroid. And in addition, a PIK3CA hotspot variant has been detected, which again can be seen in different cancers. These variants may be eligible for targeted therapy in certain indications. And then in the main body of the report, as I mentioned, there's a lot more information on potential treatments or trials in different cancers, and this can be referred to. So as in this example, is the CAP example, and it just shows ctDNA can identify potentially actionable findings that may inform treatment, but it can't determine the site of origin. These findings should be correlated with clinical information and histofindings, and integrated results discussed at a regional GTAB to guide diagnosis and patient management. Next slide, please. So this is a report where no variants have been detected. However, it's important to recognise that the result is not considered negative, but is a non-informative result. And the reason for this is we don't know whether it's a truly negative result or just there isn't ctDNA present, or there are low levels of ctDNA present. So the assay can't detect variants that are possibly below the limited detection of the test. So that is why it's considered non-informative, and a tissue biopsy may be done if possible. Further in the report, it also outlines the methodology and the limitations of the assay. And one of these to point out is discrepancies between liquid biopsies results and tumour biopsy results may occur in about 20 to 30% of cases, and this can be due to both technical and biological factors. For example, as I mentioned, there may not be ctDNA present in the liquid biopsy sample, but a variant may be picked up in the tissue sample. Conversely, variants may be detected in ctDNA that are not in the tissue, possibly due to a low percentage of tumour cells in tissue or tumour heterogeneity. And also there may be technical factors because the NGS panels will differ in the genes or the gene regions that they cover. Next slide, please. Another aspect to consider in ctDNA testing is that given that it's a blood sample being tested and the gene panel does include some cancer predisposition genes, pathogenic germline variants in these genes can sometimes be detected. However, just to point out, the assay is not designed or validated to detect germline variants, and that is not its intended use. But when pathogenic or likely pathogenic variants are detected in CPGs at VAFs greater than 30%, they will be reported as per guidelines that clinicians are alerted to these. So in this example, it's a lung cancer example where an actionable driver has been detected at 16%. But in addition to this, BRCA2 pathogenic variants been detected at VAF almost 50%. As this variant is suspicious of being of germline origin, this is noted in the report. And in these scenarios, patient consultation with a clinical geneticist is recommended. That germline testing can be arranged if deemed appropriate based on the VAF and the complete clinical picture the clinician has. And finally, it's recommended if there's any uncertainty around any of the results that they discussed at a GTAB. And next slide, please. And this slide just shows, it's the final slide just showing how the report that is emailed out will appear. So our report from the software is embedded into our EPIC report. The report from EPIC has the patient details followed by a list of the variants, and the overall summary of the results is under the molecular interpretation section. This may be provide all the information required, but then further down the report, the software generated PDF report is embedded, which has the additional information with its further detail on the variants detected and any potential trials and treatments that you may want to refer to. And this report is then emailed out to all those detailed on the request form. Thanks. I'll just hand you back to Natalie, I think.
Thanks, Sian. Natalie, do you want to quickly talk about your practice?
Yeah, sure. So basically, the other thing that I want to mention today for anyone that's interested in Cancer of Unknown Primary is we do have an NHS Futures webpage now, which is the kind of home of the UK Cup group as well. We did mention this at the recent UK Oncology Forum. If you scan this QR code, this will take you to it. You have to register with NHS Futures, but I think there's a lot of information now being put on NHS Futures. This has details. This will have details of all the educational seminars that we run in CUP, and also the genomics information and the slides, and also kind of future funding applications as well for CUP. So everything that hopefully you'd need to know will be on there alongside extra information that you might want to know about CUP. So please do join the NHS Futures webpage.
Thanks, Natalie. Terry, has Terry joined?
Yeah, I'm here.
Yeah. Terry, do you want to
thanks.
quickly talk about the national stuff that we're doing?
I will indeed. Yeah, no, thank you. And thank you. That was a beautiful overview of ctDNA generally, and it's really exciting to see this indication coming through. What I would say is Sian has already mentioned a lot of technical terms, and I'm sure, there's lots of expert people in the audience, but there's probably some people that are finding all of this a little bit daunting. So in the genomics education program, in collaboration with the various GMSs that are involved in testing and rolling out of this, we have developed, or are in the middle of developing, I should say, an educational resource targeting people on the ground that are trying to, action these tests. So like organising in the first place, deal with interpreting the results and actioning them accordingly. So if you are interested in signing up for our fully online, fully self-directed, asynchronous e-learning pack.
You've gone on mute there, Terry.
I don't know how I managed that. Maybe it's you, Paul. This is what you want to do. Find the magic button to shut me up finally. But what I was going to say was we've developed this fully online asynchronous learning package that covers all of the aspects that Sian and Natalie have just described. So looking at the indications for testing, going into a little bit more detail about why those steps that Sian mentioned are so important, like why does it matter why, what bore needle you use, how does that impact your result, why a test result might not be informative. Some of that's related to the technical stuff, some of it's inherent to the cancer biology, some of it is stuff that you have the power to control actually in terms of how you take the sample and all of that. So if you are interested in signing up for that program, you can sign up to the link that I've just put in there. We also have a wealth of resources on our G-Notes pages. So I'll just put a link in there to G-Notes, which that's the link to the ctDNA resource, but we have hyperlinks out to various other resources that might be helpful as well. So I'd really encourage you to just keep an eye out on those various links, because there will be lots of educational resources coming through. And importantly, if you do sign up and you notice that there are gaps that you think we could potentially fill, please do let us know, because we are, we have a team in the background that's hoping to develop stuff to support the wider community in rolling these things out. So do let us know.
Cool. And lastly, Natalie, anything you want to say on the GTABS access for sort of future use?
No, so I think that someone just put a comment on that actually, which I'll read out if that's helpful. So
go for it.
Genomics Tumor Advisory Board, or GTABs, have been recommended a few times, but many will not have used or accessed GTABs before. In practice, will there be guidance for individual trusts on which GTAB they access and how? So just to comment in general, obviously, there are various GTABs that are running across the whole of the UK, to be honest, at the moment, and it is a bit confusing. I know that Paul and colleagues are probably doing a bit of a survey at the moment to try and find out what GTABs running and where. We need to do better at ensuring people know which GTABs to go to. I'm sure Paul can comment for this region. For the CUP specifically, these are complicated patients, but many of the people at GTABs will have had experience with looking at these profiles and also looking at tissue of origin potentially through that. So it would be your local GTABs at the moment, and obviously, I guess Paul, you can comment on which ones people go through here.
So yes, and Terry, feel free to jump in, because I know you go to your own GTAB. So for North Thames clinicians, there is a weekly GTAB run by the Royal Marsden. So we will. So in the FAQ document that Sian mentioned earlier, we'll be kind of detailing out how you access and you can get cases onto those GTABs. I'm also trying to do that, to collect that for the Southwest and the East region, because obviously, as Natalie mentioned, you would need to go to your regional ones for your regional patients. But I think, Terry, correct me if I'm wrong, it's a case of just emailing through, isn't it, to the MDT coordinator, your particular patient, if I'm right.
Yeah, exactly. I've just popped the email for our GTAB coordinator in the. Now there is a pro forma, but if you email Ian, he'll send you the pro forma, like the main crux of what we need, because we'll have the reports on our system because they're all done in our lab, which is easy for us in our region. But it's the clinical information that's really critical. Like obviously a lot of these will be cut, but you'll have some sort of useful information from IHC or from the clinical picture or what have you. Having the clinician on the call is extraordinarily valuable, just to give that kind of clinical context, and also so that we can have that bi-directional kind of communication about what our interpretation is. We have very clever people like Sian on the call that can help provide some of the technical information behind the testing as well. If there are incidental findings like the query germline stuff, like I and some of my colleagues from the germline side will be on the call as well. And other things, we haven't even got into the whole issue about clonal hematopoiesis or other incidental things from, bone marrow related genetic changes that might crop up as well. So we can deal with all of those kind of different types of results. So we're also very happy to see some of the more straightforward cases as well. So even if you think there are things that you can manage, if you want to just bring them through just so you get used to dealing with the reports, figuring out what does and doesn't need to come through that forum, we're more than happy to use it as a kind of an educational forum in that regard as well.
Cool. Thanks, Terry. And just go back to your point, Natalie, about surveys. So within the Network of Excellence, the Circulating Biomarker Network of Excellence, we will be shortly coming out to try and survey what's happening across all regions of GTABs, but we're also looking at setting up a pilot national GTAB, which will be sort of complementary to the regional ones. So watch this space on that, because you will be able to potentially refer those patients into this national GTAB. That will probably start around September. So for between now and sort of September, I would strongly advise you go to your regional ones until we've got that set up. But more details will come out about that, because that will be applicable to kind of the wider cancer community, not just CUB. So great. I think that was probably the end of the presentation. I think any other questions for the team? I know there's obviously a few gone in the chat, and I think thanks for responding to those, Natalie. Has anyone got anything they want to particularly to raise at this point? I think what we will do is we will get the slide deck out. It'll all be available on the NHS Futures pages, including the recording. We hope to get the FAQ documents out to all the clinical teams prior to or around the date in which it goes onto the National Test Directory. And I think it's probably just worth a reminder that obviously for liquid testing, all three of your GMS regions will route into the North Thames. And then obviously if you're doing tissue testing, you do need to go through your normal, your regional GLH hub. So East and Southwest clinicians, you'll need to route that through. So if you've got any queries around that, you should get in contact with your local GLH. Hold on, we've got some questions coming in.
There was a question around, I can't answer because it's not my region, but timeline for tissue NGS. Is there a timeline for tissue NGS?
So that's West Hertfordshire, so that would sit in the North Thames. Do you know what that is, Sian, currently?
The turnaround time?
Assuming that's what it means? Is that what it means?
The turnaround time will be 14 days. As far as I'm aware, I don't think the INHS has indicated anything different on this.
Is that your question, Andrew?
Yeah, is that the question?
Andrew?
Yeah, presumably it's the timeline.
Sorry, just bringing you in. What you mean?
Sorry, the thing is, because it's so important with these patients that we get the result sort of in time. Because before I had emailed and asked about, and I was told it takes months for the patients, for these to come back, and which defeats the purpose entirely. So I wanted to know how long will it take to come back if you're running kind of, NGS on the patient for purpose of CUB where we don't have any primary.
Yeah, so from the moment the lab received the tissue, we would have 14 days, or we would aim for 14 days turnaround time for either DNA or an RNA panel, whatever is submitted, or both. I think in the past, when it was not on the test directory, it's been a bit more complicated to manage, but that's the plan.
And thanks. And can we send both, like the blood as well as tissue at the same time? Is that the right thing to do, or?
No, we would. So basically, if the patient, so if you've got a patient with that's been discussed in NDT CAR, then you would normally, if they are not on treatment, you would normally think about doing ctDNA, unless they've got lots of tissue available as well. You would do one or the other, essentially. And certainly from the studies that we've done, we've used ctDNA usually first, because it's easy just to take a blood sample while they're in clinic. But there is an option to do one or the other, but not both at the same time, because you're basically doing a similar panel of genes. So there's no point in doing both at the same time. And what I would also say is, obviously, the results will be turned around as quickly as possible. But if you've got a patient in front of you that needs urgent treatment, and obviously if they've a cut, then you would carry on with that treatment, which is presumably double chemotherapy. And obviously then you would interpret the results, and if they have a redefined tissue of origin, then that's fine. You can look at it that way, but I wouldn't hold off treatment waiting for results.
Can we send the paraffin block?
For the tissue testing, yes.
Yeah. Okay, great. Thanks.
Great. Another Mikhail question. I think Alexander's asking if we can use Order and View for CUP.
Yeah, so for those that are already set up, we use an Order View. Yes, Order View will also be able to use for CUP. I think that we've slowed down the rollout of the system, so I'm aware that not everyone within the region has been set up, but for those that are already using it, yeah, it will be available for CUP as well.
Cool, thanks. And then Richard's got a question, Natalie, around the differences between ctDNA, the two M codes. Do you want to take that one?
Can we go back to the gene panels? So essentially what we had to do was we had to look at what was available across all the different genomics hubs, and the tissue panels and the ctDNA panels are different. So we've tried to basically incorporate as many of the genes as possible that are available across all the hubs. So for ctDNA, it's got to be available across all three, and then across tissue, it's got to be across all seven. I think is the seven. So essentially, our hope is that this will be expanded. They are different panels, and tissue is a bit more comprehensive than ctDNA at the moment. But the hope is that these will be expanded as we go forward. So there are things missing in ctDNA that I would like to see, but again, it's got to be available across all the hubs to ensure equity of access.
Thanks, Natalie. Any more questions from anyone? Feel free to put your hand up or in the chat. Otherwise, we'll finish it there, I think. Okay, brilliant. Well, thank you all for joining, and